Tigadan 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 pam pam tigadan 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 pam pam tigadan 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 pam pam tigadan.
Our next presentation comes from Greg Bowman, Associate Professor and Folding at Home Director. It's going to provide a great presentation of Folding at Home's response to COVID-19. Greg, how are you today? Thank you for joining us.
Wow, thanks, appreciate it. Let me get my screen shared here. Give me one moment. All right, can you guys see my screen now?
Perfect.
Okay, perfect. So a little bit different than some of the other talks, but some of what we've been doing with high performance computing in a sort of a non-traditional way, I think it would be fair to say. So my group is very interested in protein dynamics as our major driving interest, and how understanding those dynamics can inform drug discovery, for example, and the design of new proteins. And since I imagine that many of you are not protein aficionados, I thought I'd give just a very, very quick crash course on what this is. So proteins are these little molecular machines that are responsible for most of the active processes that we associate with life. So they're responsible for muscle contraction, they're responsible for sensing light in the eye for vision, they're responsible for sensing vibrations in the ear for hearing. They digest your food and they build new biological components. So they're busy little machines. So these things are made out of chains of chemicals called amino acids. So here's an example here just showing the different atoms that are in an amino acid. So nitrogen, carbon, hydrogen, oxygen, and so on. So all of them have this common backbone element, we call it. And the nitrogen of one amino acid is linked to this terminal carbon of the next in a linear chain. And then each is decorated with one of 20 chemicals, typically called side chains, that's represented by this R. And the upshot is that the ways that these atoms push and pull on each other allow many proteins to spontaneously fold into reasonably specific three-dimensional structures that are capable of performing some function. So I won't go into detail on this specific protein. I'll just point out that this is what we call a ribbon diagram. So this ribbon is tracing the chain of amino acids that make up this protein. So you can see the two ends here and here. And this ribbon just helps highlight some of the common structural elements, like these helices, as we call them, which are these barber pole looking things, and beta sheets, which are depicted as these big long arrows, and then loops in between. These ribbon diagrams are very convenient for getting a sense of the topology of the protein. You can see down here that the size scale is quite small, about 5 nanometers. So these are beyond the detection limit of any conceivable microscope. So there's no way to watch these things in action. One downfall is about these depictions and these ribbon diagrams is that they can give the false impression that there's a lot of empty space. So here's another depiction where I'm showing a little sphere for every atom in this snapshot of this protein that highlights that this is really a very compact shape. Right here, you can see that there's a concavity in this protein surface, and that is what we call the active site, because that's where this protein actually does something active. It catalyzes, enhances a chemical reaction. So one of the challenges with this is that experimentally we can just get these single snapshots of proteins, but like the machines that we're used to working with on macroscopic scales, these things have lots of moving parts, and we're just blind to that. And like I said, there's not currently even a conceivable way that we could experimentally watch how every atom in a protein like this moves over time. And so since the 1980s, I would say, one goal of computational biophysics has been to use computer simulations to watch these processes. So here I'm showing a depiction of the same protein, sphere for every atom. Now they're just colored by the atom type, so whether it's carbon or nitrogen or oxygen. And what I'd like to show is just a snippet of a movie where we've been running one of these molecular dynamic simulations where basically all we're doing over and over and over again is asking how each atom in this protein and the surrounding water and other surroundings push and pull on each other. And based on that, we can keep asking, where is each atom going to be some small time in the future? And you can see all kinds of interesting motions happening. So here in the part that I've highlighted with the label cryptic pocket, you can see that there's this opening of these structural elements called helices, creating this big groove that is comparable in size to the active site. And we never would have guessed that it was there based on the initial structure that I showed you, the snapshot from experiments. It's only when we get to watch the dynamics of this thing that we can find features like this. And it's super exciting because apart from this information from the simulations, the only way to drug this protein is to try to find something that binds to the active site and blocks this protein from performing its job. Right. And so that's very useful if you want to block the protein, but there's all kinds of problems because there's many proteins that do similar things and you might want to block one, but not the others. And that's hard if their active sites look essentially the same. And there's many cases in human disease where you have a protein that has malfunctioned, right? And so you don't want to block its active site. You want to make it work better. And so these cryptic pockets, as we call them, because they are hidden to us without the simulations, present all kinds of interesting opportunities for specifically targeting proteins without getting off target effects or enhancing desirable activities, or just finding new ways to target proteins that are difficult drug targets. So one challenge, though, is that these simulations are extremely computationally expensive, right? So this movie here took, weeks on take an order of a week on a desktop computer. It's very short, actually, in terms of the time that we've simulated and getting to the sorts of like milliseconds and seconds timescales that we'd like to access to connect with experimentally and biologically relevant timescales can easily take anywhere from 100 years for very simple problems to tens of millions of years or age of the universe type calculations, on a powerful desktop computer. And so, one can get much further, of course, with high performance computing, getting many computers working together, but we're still limited, in fact. And people have even built special purpose hardware just for running these simulations, and we are still limited by data acquisition. And so what my group and a number of others have been doing is saying, okay, well, it's extremely valuable to take what I'm going to call a computer science approach to this problem, getting more computers working together efficiently on one calculation. But there are also interesting opportunities for bringing in more physics, right? And so one of the things that we're very interested in doing is saying, okay, well, let's not just think about this as watching how every atom in a protein moves over time, which is the stochastic process. Let's think about this as building a map of this space of protein conformations. So our approach to this is called Markov state modeling, or MSMs. And the idea is that, the nodes in these networks correspond to structures that are reasonably probable for the protein to adopt. And what we want to do is use our molecular dynamic simulations to learn the identities of these states and the rates of hopping between connected states, right? And this is an extremely powerful framework because we could build one of these networks by running one long simulation that stochastically hops around the space and eventually gives us data on the behavior of each of these states and how fast each of these transitions are. But from this perspective, it doesn't take too long to realize, well, you could get that data from one long simulation, but you could get it much more quickly in terms of wall clock time if you instead ran many simulations in parallel. Ideally started from different initial structures and let them long enough to explore locally. And as long as they overlap with each other in the space they've explored, you can now piece this together into a more global map that captures processes on length and timescales that are far beyond the reach of any individual simulation. So just by analogy, this is equivalent to saying, okay, well, I want to build a new maps app of the United States or Europe or whatever, right? And so you could say, okay, well, I don't have satellites, so I'm going to drive around and find all the cities and roads and put together this map, right? And so you could do this by building the fastest Formula One race car you could and zipping around and trying to build this map serially. But we all know it would be much faster to build this app by getting a lot of people to send you their GPS coordinates at regular intervals in time as they commute to work and make a run to the grocery store. And as long as they're exploring, somewhat different but somewhat overlapping spaces, now you can piece all of this data together and extract this map. So with this in mind, we can say, okay, well, now this gives us a framework for integrating data from arbitrarily many simulations. Of course, funding agencies aren't going to give me, my group, $1 billion to build our own supercomputing facility that dwarfs everything else. And so the approach that my graduate mentor Vijay Pandit took was to start a project called Folding at Home. And the idea was to invite via the internet anyone with a computer and an internet connection who wants to become a citizen scientist and to dedicate a portion of their personal computing power to running computer simulations for us in the background. And so there's a lot of room for people to vary their levels of participation. You can say, how many CPUs and GPUs on your machine we can use 24/7 in the background. You can install our software and set it up so that it only runs when you've been inactive for a certain period of time, and it pauses as soon as you start using your computer so that it's never in your way, and so on and so forth. But the idea is that you install our software on your personal computer, and what it does in the background is it reaches out to our servers and notifies them that you're available to do work and what hardware you have available. Again, which is your choice, how much of your computer you donate to us at any given time. And then we have queues on our system for simulations, and so you'll get assigned at random an appropriate work unit, we call it. So you get told, here's the starting structure and the parameters for the simulation and how long to run. And then your machine goes off in the background and runs the simulation. So keeps asking where each atom is going to be some small time in the future over and over again, and stores the structure of the system at regular intervals. And then once you're done, you send the data back to our servers and you are awarded points in proportion to how hard the calculation was and how quickly you did it. And so it's super cool. People can form teams. There's a whole ecosystem that's grown up around this. People have created cryptocurrencies that distribute coins based on points that you earn in folding at home in a given period of time. People have done detailed studies of what hardware to buy to get the most points per dollar spent per day. It's a super engaging community. And so at the beginning of 2020, before the pandemic, we had about 30,000 active devices around the world participating in this project. And then we celebrated our 20th anniversary this past fall and have generated hundreds of publications. And just before the pandemic, where we helped put a antiviral drug into clinical trials. So we've gone all the way from very basic research on this protein folding problem. So how do these proteins fold up on themselves into these functional structures, to very translational work in the drug discovery arena. And we've learned a lot about what these spaces look like, these maps look like, and that has informed the development of new algorithms that I think could be appealing to this community because they allow a level of parallelism that's quite impressive, since we can scale to arbitrarily many computers, and because we're building these maps on the server side, if a simulation goes down, it slows us down a teeny tiny bit. And of course, the more simulations we have, the better, but we're largely insensitive to failure. So we can scale without issue to arbitrarily number many computers. And one of the things that's proving really cool recently is our work with what are called adaptive sampling algorithms. And so the idea here is that instead of just letting a bunch of simulations go for some fixed period of time, and then collecting all the data at the end and building one of these maps, we're going to take a more active role. So we're going to run one batch of simulations, then we're going to build a map from all of the data that we've generated so far. And now we're going to use this map to decide where should we start the next batch of simulations to make the most effective use of the compute power that we have available to us. So this is just a little toy. So we've got this space here where blue is low energy or high probability conformations, we'll say, on this grid. And then moving up to red is high energy or low probability, right? So our protein is going to want to spend more time on grid points that are blue, exponentially more time than on grid points that are red and are essentially inaccessible. And just to mimic the sorts of problems we work on, we can pretend that we have a single snapshot from experiments, right? So we have this starting point right here. And, as is often the case, we have a specific thing that we'd like to see, right? So oftentimes my group is hunting for these cryptic pockets to find new ways of drugging proteins. And so one of the heuristics we're interested in is finding structures that have larger pocket volumes, which we'll say is to the right, than smaller ones, with the idea that something has opened up, potentially a cryptic pocket over here that might be an interesting target for drug discovery. And so given that we can't thoroughly explore all of this space, it's way too large, what we'd like to do is dedicate more of our compute power to exploring regions over here that have the sorts of structural features that we're interested in. So what we're going to do is start our batch of, say, 10 simulations, in this case from our starting point here. We're going to let them explore locally. And so in this little movie, I'll show the sort of light green or yellow dots are where these 10 simulations are at the moment. And then we'll cover color regions that have been visited by the simulations in dark green. And so what we're going to do is, let this batch of simulations run locally. We're going to build one of these maps, and then we're going to rank all of the states in this map based on this function over here that has two components. One is a geometric component, so it's going to favor states that are to the right over states that are to the left. And so that's going to favor exploring regions over here. And the other is a statistical component. And what that's going to do is just favor broad exploration as opposed to redundantly sampling the same states over and over again. So it's going to favor states that we have little data for over states that we have lots of data for. And so when we start these simulations, what you can see is that they explore randomly at first. Then you can start to see what looks like rightwards progress. They get stuck here because they're surrounded by these high red, low probability regions. But then they're able to backtrack and make significant progress to the right and really explore this space. Just to contrast that with what we would get if we perform the same amount of simulation without the adaptive component, we can do a little erase here. So at the top is the same movie I showed you before. At the bottom, we're just going to run 10 simulations and let them go doing their stochastic random walks around this space. And so what you can see is at the very beginning, they don't look so different. They both go over here and get stuck. They both happen to back up. But then these long conventional simulations go the, air quotes, wrong way from the perspective of what we're more interested in. And they just keep redundantly sampling the space that isn't relevant to the problem that we want to solve. Whereas we make very effective use of the finite computing resources with our adaptive algorithm. So our favorite way to run things at this point is to run some adaptive sampling on our cluster locally. So we get some initial exploration of this space. Then we perform a clustering of all this data. So we build one of these Markov models, and we go and shoot off a handful of simulations from every state in that model to make sure that we get reasonable sampling of each state and have the statistics that we need for each state to build a robust and reproducible model. So we were going along, we're doing a lot of work on Alzheimer's disease and multiple forms of cancer and Ebola virus and all kinds of different biological problems. When at the end of February, it became clear that the SARS-CoV-2 virus and the corresponding COVID-19 disease were going to become a pandemic. And given that we have this very general platform that we were already taking in many directions in parallel, what we did is dropped everything and put all of our intellectual and computational resources to bear on the proteins that allow the SARS-CoV-2 virus to infect a host and evade its immune system and replicate the virus, looking for, trying to understand how these little machines work and how we can either shut them down with therapeutics to treat infected patients or potentially inform the development of new vaccines. Because of course, when we jumped into this, the vaccines were not available yet. And so we put out a call to action to help out, and hopefully the sound will work here.
You can help fight COVID-19.
The pandemic felt so overwhelming. Staying at home wasn't enough.
The novel coronavirus has humbled every nation and every individual.
But it doesn't have to be like that.
Maybe if you could, gather all the computers in the world and put them to work.
And to be able to do things that you could not do by any other means.
With your help, we can.
Folding at Home is using its distributed computing model to create the most powerful supercomputer on Earth.
The collective power of millions of volunteers to aid COVID-19 research efforts.
And a grassroots example.
Your rig is helping researchers find treatments and vaccines faster.
We need people like you.
Working together can make such a big difference.
You can learn more at foldingathome.org.
And so what happened is that, we started our first SARS-CoV-2 simulations at the end of February. Like I said, we had about 30,000 active devices at that time, which was allowing us to perform multiple calculations per year that would have cost millions of dollars on the cloud. That we thought we were in a great position to help. It turned out that many people saw our calls to action and agreed, and lots of people were feeling helpless, being stuck at home and not being able to do anything to take action to improve their situation. And so we had explosive growth in Folding at Home. So here in blue, you can see the cumulative number of downloads of Folding at Home over the first couple of months of the pandemic. In orange is the growth in the number of COVID-19 cases. And so you can see that within the first couple of months, we went from having 30,000 active devices, which is on the scale is essentially nothing, to well over 1 million devices running around the world. And so, this was quite the wild ride. We are our academic, background, mostly we're using small portions of our grant funding to buy some servers, which are hosted at our universities. And so, scaling up to meet this demand, we were, our architecture was well suited for scaling. So we handled the first 5-fold growth without breaking a sweat. But as things got to 10-fold growth and beyond, we started to, slowly grind to a halt. And so, with the time scale for applying for and getting grants and buying hardware, it would be like a year, at least probably a year and a half. So that wasn't going to help. But fortunately, with having been around for 20 years, we've had, passionate volunteers kind of spread around the world, and many more who joined them in their desire to help with work on COVID-19. And so we had all these companies reaching out, trying to see how they could help. And long story short, we're very grateful to many who helped us to quickly get onto the cloud and scale up to harness all of this compute power and bring it to bear on the SARS-CoV-2 virus. And so the upshot is that for a while we became, by far the most powerful computer in the computing resource in the world. Even with a very conservative estimate of the compute power of each of our volunteers, we were the first to reach the Exascale and had at least 5 times the performance of the Summit supercomputer that was the fastest in the world at the time. And I think this is probably a gross underestimate, and we're working on improving the estimate now that things have calmed down a little bit. But coming to the virus, I thought I'd tell you a little bit about what we actually did with all of this compute power. And so with all of this, we actually simulated basically every protein that we could from the virus, which is quite a transition. In most cases in our field, people will simulate one protein for about a microsecond. So that's a standard, reasonable paper data set for a paper in our field. With all the compute power that we had, we generated about 0.1 seconds of data for our first paper. So that's 100,000 times more data than anyone is working with. And had like milliseconds of data for each individual protein. So 1,000 times more data than in a typical paper per protein for many different proteins. And one of our focal points was on a complex of 3 proteins called the spike. As you're probably aware at this point, each of these red protrusions on the surface of the virus is a spike. And these are incredibly important because they are responsible for binding to a potential host and initiating infection and entry of the virus into the host. And so because of this essential role and the fact that they are exposed on the surface of the virus, they are very attractive targets for therapeutics. They're the main inspiration for the mRNA vaccines that were the first to get approved and put into patients. And they're also a big susceptibility for the virus because they're the main thing that our immune systems have a first crack at recognizing and mounting an immune response to kill an infection before it gets out of control. And so the virus has evolved a number of really fascinating defense mechanisms. The one that we focused on is called conformational masking. And so the idea here is that if we look at one of these spike complexes, here it's made of 3 proteins, so they're each in a different color. These ribbons trace the chain of amino acids in each of these proteins. And the transparent surface is just a space-filling representation to remind you that this is a very compact structure. And the green is sugars that decorate the surface of the protein, or another defense mechanism that I won't go into. So the bottom of this complex is where this thing is tethered to the surface of the virus. The top is where it's going to be potentially binding to a protein called ACE2 that is on the surface of potential host cells like many human cells. And so one of the things that this thing does is that this closed or down structure, it is the most common structure of the protein that we've seen, that's actually closed up on itself. So the surface that is responsible for binding ACE2 is buried in this structure, right? And that's kind of the equivalent of a turtle pulling its head into its shell, right? It protects the most vulnerable part of the turtle, its head, but it also means that the turtle can't eat, right? It can't attack its prey if it's got its head pulled into its shell. And so in this conformational masking strategy where the protein is mostly closed up, it still needs to be able to open up to actually expose that key surface and bind to a potential host. In the early days, people didn't really know what that open state looked like. Some of the early experimental data captured what was called the up state, where this orange part of the protein has poked out just a little bit. And so people have thought of that as the structure that is responsible for binding ACE2 and initiating entry of the virus into a host cell. But in looking at this, we thought, no way, like it can't just do this. There are so many contacts broken that this thing is probably flopping around. There's a good chance that this thing opens up way more broadly, and we just have no idea. And characterizing this heterogeneous open state, if we were right, is really valuable because, again, if you only know about these 2 structures, there's some opportunities for drug design, for example, but you might be missing a lot of the story and a lot of potentially better opportunities for therapeutic development. And so here we thought of this as a Demogorgon monster because you have these threefold symmetry, and somehow this end of the protein has to open up. So we envision this as being like the mouth of one of these Demogorgon monsters from the TV series Stranger Things. So in this orientation, the upper left is where this thing is tethered to the virus. The lower right is where it has to open up somehow. Many people have run microsecond timescale simulations of this thing and just seen it wiggle around very subtly. But with the power of our adaptive sampling algorithms, followed by extensive simulations unfolding at home, we were able to capture much more dramatic motions of this system. So here you can see this part of the protein opening up quite broadly. So you've got this huge gaping hole. Here's the surface that actually binds to ACE2. And what you can see is that that surface and many others are exposed by this opening. And so this is really interesting because in the closed state, you wouldn't think that anything in here is a viable drug target, for example. But once you know about this open state, it's very interesting to think about targeting these regions. And in fact, it turns out that retrospectively, some of the early antibodies that were discovered that targeted this protein do bind in these deeply buried cryptic sites, as we call them. And so this, has been corroborated by experiments now, because no one else could explain how an antibody could get in here and bind these deeply buried sites. So this is super exciting and provides a basis for doing all kinds of other things, right? So we also simulated the spike complexes from 2 other coronaviruses that also bind ACE2 to initiate infection. These are the NL63 virus and the SARS-1 viruses. And so what I'm showing here is the probability on the y-axis of different extents of opening of one of these receptor binding domains, or RBDs, like we saw opening up in the simulation that I just showed you on the previous slide. And here's some representative structures showing the extent of opening that these different points correspond to. So all the way out here is very rare, but very open. Whereas over on the left is very closed up. And so what you can see is that the NL63 virus out of those 3 is the most closed. That's very interesting because the NL63 virus was only discovered when people went looking for coronaviruses after the SARS-1 outbreak. I think it's quite widespread, actually. I'll have to fact check that, but it's associated with symptoms like the common cold. And so what we would predict from this is that it is very good at evading an immune response because it's closed up so often, but it's actually quite bad at infecting potential host cells because it doesn't open up enough to be likely to infect a host when it approaches a host cell. In contrast, over in red here, you can see that the SARS-1 virus is by far the most open of the spike complexes. And this is really interesting to us because the SARS-1 virus was the most deadly of the coronaviruses that we know about. 1/3 of the confirmed patients with SARS-1 died. So a very high mortality rate, but it didn't spread nearly as much as the SARS-2 virus, for example, because we think it was quite susceptible to an immune response. So if your immune system failed to respond adequately, you had a high chance of dying, but lots of people's immune systems, we think, were successful in fighting off infections in the early stages, so this thing didn't spread as much. And there's some other factors to consider, so please don't think I'm oversimplifying things, but at least to a reasonable extent, it's accurate to say that the SARS-2 spike is in a sort of Goldilocks scenario, where it's far better at evading an immune response than SARS-1 by being more closed, but it's significantly more open than NL63, so it's far more successful at entering into host cells than the NL63 virus, for example. And as we all know, this has enabled the SARS-2 virus to become a pandemic that has really, done a lot of damage. Words, I don't have the words, but I don't need to tell you. So in addition to looking at, all these different versions of the spike, we've also done at least 60 other systems where we've looked at different SARS-2 proteins. We've looked at their homologs and other coronaviruses to understand, what's special about SARS-2. We've looked at a bunch of human proteins that the virus interacts with. As I said, in total, even in our first paper, we generated over 0.1 seconds of simulation data. We probably have another couple tenths of a second of simulation that is unpublished so far, which is just astounding given that, like I said, most papers have a microsecond of data. So we've got hundreds of thousands of times more data. It's quite large, over 200 terabytes. And so, we're always looking to share our Folding at Home data since it's publicly generated. But normally we've had to do that by, mailing hard drives to people upon request. Fortunately, there's some new services that help. So we've got our Markov models, these maps we build up on a website called OSF. I think it's Open Science Framework, but don't quote me on that one. And we partnered with the AWS Public Datasets program to share the raw simulation data for those who are interested in that. But we generally suggest that people work with our maps since they're far more compact and easier to work with. So I won't talk about all of the systems that we've worked on, but just to show you, one other example, very briefly, we worked on a protein called NSP16. This is another essential component of the virus for evading an immune response. For the sake of time, I won't go through the details, but suffice it to say that this protein catalyzes a chemical reaction here with these two small molecules, these two substrates, but this reaction is also performed in humans. And so attempts to design drugs that bind the pockets that these two molecules are in have found drugs that can inhibit the activity of this viral protein and hypothetically slow a virus down and give our immune system time to kill off an infection in the early stages, but they end up not being useful drugs because they also hit the human proteins, and so they have significant off-target effects that are quite bad for patients. And so, long story short, we have some cool analysis methods where we can take simulations of proteins under different conditions. We run them through an autoencoder to coarse grain the data. But importantly, in addition to requiring these autoencoders to take a protein structure as input, collapse it to a low dimensional representation, and then be able to reconstruct the original high dimensional input, we also require these networks to predict a biochemical label. So, is the viral protein more or less deadly, or is the enzyme more or less good at catalyzing some reaction? And that helps us to really be able to identify what the important structural determinants of these differences between proteins are. And so again, skipping over some of the details, we found this very interesting cryptic pocket in this protein. You can see in orange the parts of the protein that are buried in all of the structures that people have solved experimentally, but that get exposed when this motion occurs. And we think this is super interesting because this motion does not occur in human proteins, related human proteins. So if you target it with a drug, you won't impact those human proteins, just viral proteins. And this pocket is shared in the SARS-1 and MERS viruses, and we think based on sequence comparisons, all of the extant coronaviruses that have been sequenced. So targeting it could provide a way to develop a pan-coronavirus inhibitor that could be useful in the current pandemic, but also against future coronaviruses that might threaten humanity, which is basically guaranteed to happen, right? These things aren't going away. And so, it's super exciting to be able to get at this information with the simulations. I haven't talked about it, but a third of my lab is experimental. So we actually are doing drug design and have targeted some of these pockets in other proteins we've worked on and showed that we can control them in other ways. But for the sake of time, I'll just say thank you to my lab and the other labs that are involved in the Folding at Home project, to all of our volunteers around the world, our funding sources, and all of the companies and other organizations that have come to our aid to help us make, like I said, the most effective use of all the compute power that's been made available to us to get science done as quickly as possible. With that, I'll be happy to take questions.
Thank you, Greg. I currently don't have any questions in the queue. Hussein, do you have a question? You seem to have one for each session. So
it's very interesting. I really enjoyed this presentation. It's a comment, actually, not a question.
It sounds good. Thanks.
Yeah, really, I really enjoyed it. And could you tell us about the platform? I mean, all these simulations that you have presented, what kind of platforms they are running? I mean, is there a benefit from accelerators? I mean, from the HPC world?
So we make a lot of use of GPUs, the technology that is useful for updating every pixel on your screen in parallel is also useful for updating the position of every atom in a protein in parallel. So those are a huge boon for us and where most of our compute power comes from at this point. We're set up to work on both NVIDIA and AMD GPUs. So both are extremely valuable contributors. I would say the largest portion of our volunteer base is gamers who have, invested in powerful computers and, want to save the world virtually. And so they're excited to use their compute power also to help save the world in actuality. And so, we do make a lot of use of GPUs, but we also run on CPUs as well.
Excellent. Thank you.
So last time I checked a week or two ago, we had about 50,000 GPUs actively participating in the project and 150,000 CPUs. But of course, most of those are multi-core. And so there's still quite a lot of compute power. Again, conservatively estimated at about the same performance as Summit's peak performance, but again, very conservative. And our algorithms are really interesting too. So there's a lot of interest, from more traditional HPC folks in our community of late, because they're looking at how they're going to run molecular dynamic simulations on exascale supercomputers as they come online. And of course, once you go to that scale, you're pretty much guaranteed to have failures, node failures and hardware failures on a regular basis. And so this is a really cool way of being tolerant to that, because like I said, it's only a little bit of an exaggeration to say that we don't care if one simulation goes down as long as the vast majority of them are running smoothly. And so, it's a super powerful way to go to scale.
Yeah, thanks, Greg. And Hussein, one of the 13 clusters we have at the Council Center, I believe it's a V100 base, and that was one that we dedicated at least for a couple months for the Folding at Home project. While this was happening.
Appreciate it.
It was. We're more than happy to help, and it was at least one system we could spare during the overall process, because our clusters are used almost 24/7.
So this is one question that I'm curious about, is that my understanding is that when you're running these large high performance computing facilities, that you want the system to be utilized, but you also ideally would like some space on the queue for people to run. And so are you guys always at max? Are you always running at full throttle, or do you normally have some spare capacity?
There is a little bit of spare capacity. It's not much. I'm always asking our cluster center manager for additional resources for some various other projects, and then he gives me the queue of what's lined up. It's like, okay, well, get back to me when I have something. We're pretty busy.
Because I'm always curious if there's ways, opportunities to be, running Folding at Home, preemptively, for example, so that things are busy, but we get kicked off anytime people, have an active need for resources. So.
Right. Well, thank you, Greg. During this, I didn't receive any other questions come in, so leave it as that.
So usually there's a period of time where, where you can benefit from it. I mean, I don't know, Christmas, New Year, all these periods where little people actually just running on the machine. So benefit from it.
Yeah. No, that would be great. Yeah, we have everything containerized at this point, and we have a number of companies and other organizations who are just running these containers in the background, overnight when the gaming users are less busy, for example. And so that's all there for people to make use of.
Okay.
Thanks again, Greg.
Yeah.
Bum 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 bum.